bringing this home a little bit now. We've seen how this disease is observed on our landscape and how it progresses in the animal and how it persists in the environment. And so now it's time to look at some potential effects of this and interactions. So our next speaker is Amy Girard, one of our local citizens here. Amy received her master's degree from the University of Wyoming, the real UW, through the Department of Animal and Veterinary Sciences. Her work is focused on wildlife disease, the transmission dynamics of the parasite Eleophora snyderi, and also the effects of genotype on CWD-driven mortality in elk herds in Wyoming. Amy has studied a wide variety of animals and birds, but prior to grad school, she worked with Wyoming Game and Fish Department studying brucellosis on elk feed grounds. But Amy currently works for the Teton Weed and Pest Department. So, Amy?
So first off, I wanted to apologize to the museum. I brought my water in, but I think it's for everyone's benefit. So thank you so much for inviting me here to speak to you today. I haven't had a chance myself to present on this information. As Tom pointed out, this was a side project that I did or collaborated with at the University of Wyoming. And the whole point of it was to try to understand how these genotypes in elk might influence population dynamics in an elk population that use winter feed grounds. So I kind of wanted just to give you a little layout of how the talk is going to work so you kind of know where I'm headed. First off, I'm going to talk about these genotypes and the differences in susceptibility to chronic wasting disease with focus on elk. I am, of course, going to talk to you about the model, what we did and the results, and how this might pertain to what would actually happen around Wyoming. And I am going to talk to you a lot about heterozygosity and what that might mean for these elk. And I want to really talk about that in terms of my research because, so we published this a few years ago, and so I'm aware of some of the concerns that people generally have about this project, and so I'm going to try to highlight some of those in particular. So as we know, chronic wasting disease is always fatal in those susceptible hosts. But there is a lot of variation in the incubation time. So these incubation times are, again, as it's kind of been pointed out, but it's that period of time from which an individual becomes infected until we see the presentation of clinical disease. And the reason we see variation in incubation times is due to differences in genotypes. And those genotypes are combinations of potential alleles at a different or at a locus. So in elk, there's 3 different potential genotypes that codon 132, which is the gene that encodes for that normal cellular prion protein. And as it's been already pointed out, that elk have normal prion proteins just like we do. So there's 2 amino acids that make up this particular genotype. And it can either be methionine or leucine. So the way these present in these genotypes is they can either be homozygous for M, presenting as MM. They can be homozygous for the L, when be LL, or they can be heterozygous and have an have an allele of each. So there's a lot of variation in how these frequencies present in wild and free-ranging and farm-raised animals. We've always seen a predominance of the MM genotype, and it can range anywhere from 65 to 85% in free-ranging elk populations. The second most common genotype is the heterozygous ML, and it ranges from 17 to 30% in populations. And then we have that homozygous L allele, so it tends to be quite rare in the population, from 0 to just over 2%. I do want to add that these are reported from wild populations, and in farm-raised elk, we do see a substantial variation in these frequencies, with one instance where LLs were reported as high as 10%. So to try to understand how these genotypes influence incubation, we can look at some experimental studies. But as Dr. Wood pointed out earlier, there hasn't been that much work done in elk. So I'm just going to point to this one study in particular, where they fed homogenized CWD-infected brain to CWD negative elk. And what we found is that, or what we didn't find, I wasn't part of that study, but what they found was that MM genotype had a much shorter incubation time, so almost 2 years before they saw clinical disease post-inoculation. That was followed, of course, by the ML, which showed clinical disease just after 3 years. And then the LL situation is a little bit more complicated. In that study, they didn't see clinical disease until almost 5 years, and just over 5 years. But at the end of the study, the second part of that study, they terminated the remaining elk, and one of those LL elk was still not showing signs of clinical disease. There is some significant variation in how these genotypes, differences in the incubation times for these genotypes. And again, so if we look at positive CWD samples from, again, farm-raised and wild populations, that MM genotype is significantly overrepresented in positive samples. So what this means that if that MM genotype is found in your population at 60%, we would expect to see roughly 60% of your positive samples showing that same genotype. But what we see instead is that they are overrepresented in comparison to their prevalence in the population. So it's a similar situation in deer. I don't want to get into all the polymorphisms for white-tailed deer. There's a lot, and it would be just a little too confusing, I think. But we see a similar scenario play out, where certain genotypes have longer incubation times. Robinson et al. Found that older age class white-tailed deer had a higher prevalence of those genotypes that offered resistance to CWD. And there has been one genotype that's been reported in white-tailed deer that's absent from all CWD positive samples, though that genotype is quite rare in the population. And again, there is a lot of geographic variability between the frequency of these different genotypes, even amongst neighboring populations. In mule deer, it's a little bit easier to kind of get at. There's just one single allele's change that determines the incubation time in CWD positive animals. And it's that F allele is the one that has the longer incubation time. And again, it tends to be underrepresented in our CWD positive samples. And this allele varies from 0 to 22%. And again, it's quite variable by geographic area. But it's not just those cervids or animals or deer members that have these longer incubation genotypes. The same thing with people. In cases of variant Creutzfeldt-Jakob disease, they're almost exclusively homozygous for methionine, so they have that MM. And we see the same thing in sheep and goats. And so there's been a lot of intensive breeding programs to try to eliminate or reduce the presence of those susceptible genotypes. So what we wanted to do is try to incorporate these differences in genotypes, to try to understand how they might influence long-term population dynamics in an elk population that uses winter feed grounds. So that was kind of our overarching goal. And this is actually here at Elk Alive Feed Ground, my favorite feed ground. So part of the point of this is that there's a lot of uncertainty about CWD in feed grounds. We just don't know what's going to happen when CWD arrives at these feed grounds, though we are fairly certain that this is likely to happen. And part of that uncertainty evolves around the fact that feed grounds have the potential to have greater than average prion contamination. We believe that this is the case because if we look at areas where elk and deer are held in large numbers in either captive populations from research or farm-raised scenarios, the infection rate tends to be much higher than anything we've seen in natural populations. And then we also know that areas that have infected animals that congregate in large numbers, whether natural or man-made, have the potential to become prion sinks. And this is a large part because, as it's already been mentioned, these animals excrete prions in pretty much every way possible, whether it's the saliva or blood or carcasses. So anywhere that these animals are really congregating, we expect to have a large amount of environmental contamination. So what we wanted to do was try to create what, in our minds at least, was a worst-case scenario. So we know that in our research facility at Sebille, they have unnaturally high levels of prions at this facility. The first CWD case was documented in 1979, and numerous inoculation studies have been conducted there. Animals that are susceptible to CWD become infected there. They don't leave without CWD, or they never leave actually, but they don't leave without CWD in particular. And again, we thought this was a worst case scenario because animals here that are subjected to that environment never have the opportunity to leave that contaminated environment. So as opposed to free-ranging populations that might use an area that's highly contaminated, in other times of the year, they at least have the potential or the opportunity to try to avoid those areas by different means through migration or just seasonal movements. So part of our model was using captive elk data. And so 39 elk calves came from the National Elk Refuge in 2002, and they were held in CWD-contaminated holding pens at Seville, and they were routinely tested for chronic wasting disease. And then they recorded the number of days of death for each animal. So some of those animals were euthanized when they were displaying, or when death seemed imminent, and other individuals were found dead. So if we look at the frequencies of the genotypes in the captive elk data, we can see again that the MM is the most frequent, followed by the ML genotype, and then that LL. There was just one single LL in that sample group. So those actually reflect what has been found or been documented in free-ranging populations pretty closely. So the days to death for those individuals, for the MM genotype, it is, of course, shorter. It ranged from 830 days to 2,568. And for the ML, it was over 1,500 days to over 2,600 days. And then again, I put in here that the LL was alive at publication, but as Dr. Wood mentioned earlier, that LL elk is still alive, but she was treated, actually she was thrown into with the MLs, and she was treated as a 12-year-old animal. So we did use the Kaplan-Meier survival curves to try to understand differences in survival between the two cohorts. So we used the MM cohort and then any individual that had at least one copy of the L allele. And so using a log rank test, there was a significant difference in the probability of survival between these two cohorts. So again, those genotypes are influencing how long they're surviving at the research facility. The other half of our modeled elk population was fed information from the Pinedale Elk Herd Unit. I selected this Pinedale Elk Herd Unit because it's been intensively managed and there's been a lot of research associated with it. So we have pretty good data in terms of the demographics of that herd unit. So I used the age structure, which comes from the teeth aging process when those animals have been trapped. Used cow-calf ratios, the initial population size, and hunting and non-hunting mortality. The hunting mortality for this population is quite high. At the time of this analysis, the Pinedale elk herd was above objective and had been for several years at least. The non-hunting mortality includes predation and any other management mortalities. And I will just say that it is very low. These elk tend to be pretty hardy. And predation for this population, at that time at least, was very low. So non-hunting mortality is pretty insignificant, but it was included. And then we used that information and added to the probability of death from CWD based on those observed days to death from that captive elk herd. And this is modeled over a 100-year period. So we created a stochastic spreadsheet model using at-risk software. And so we fit probability distributions to those days to death based on chi-square goodness of fit testing. And then those distributions were used to bootstrap a logistic distribution to determine annual probability of death. And I'm not going to get into any more of the very specific details of the model, but it is online, it's freely accessible, so feel free to access it and email me with any particular questions. I did want to bring up two very important points that have come up a lot. So in this model, we weren't actually looking or modeling prevalence. Instead, what we did is assume that every elk is coming into contact with infected material. And so we're not trying to see how prevalence changes through time, but we're trying to create, again, this worst case scenario. So instead, we're saying that every animal has the opportunity to die from CWD based on their genotype and age. So every year that they are still alive, they have the probability of dying, again, based on their genotype and age. We also assumed that there was no fitness disadvantage to having an L allele, and I'm going to talk a lot about that towards the end of this talk. So we used a few different scenarios. First, we looked at age-specific mortality to try to understand what it would look like without genotypes influencing this population. So your probability of death from CWD was just based on your age. And we broke it down into a hunting and no hunting scenarios. And hunting was just the current hunting levels in the Pinedale elk herd unit. The genotype-specific scenarios incorporated age-specific mortality and genotype, and there was 3 different scenarios, including the current levels of harvest, antlered only, and then a no-hunting scenario. We did conduct a sensitivity analysis, which showed us that the model output was quite sensitive to CWD mortality. So what this means is that if our understanding of CWD mortality is wrong, then this is going to have a large output or a large effect than what we would expect in the model output. It also said it was sensitive to changes in hunting mortality. And again, hunting mortality is quite high in this population, so hunting has a wide range of effects in what we would expect in the outcome. So just going to jump into the results, starting with those age-specific scenarios. So these are the ones that do not include genotype. So with current levels of harvest, the population was extirpated by a year with 100, but they were, as you can see, the levels of elk were unacceptable way before that time period. In our no hunting scenario, the population dipped below objective pretty quickly, and the trend does suggest that this population would be extirpated. So then we incorporated, again, these are the the genotype specific results. So if we look at current harvest levels, the population decreased by over 80% by year 25, with 0 elk remaining at year 100. In our antlered only harvest, the population declined, but the model stabilized, suggesting that it might actually increase if the time scale was drawn out. The lambda in those last 10 years was 1.01, which lambda is a measure of population growth. So again, this population suggests that it's becoming stable, and again, maybe it would increase in the future. In our non-hunted population, it declined initially, but then by year 100 was not sustainable, and it had 32% more elk than it had in the original starting point. We did see changes in genotype frequencies. You can see at the current harvest, or the current hunting year, I'm reporting only the frequencies at year 50. At year 100, we didn't have any elk, so I'm just using year 50 for that example. And so in that current hunting, the M allele decreased by 42%. And in the other 2 scenarios, it decreased by approximately 6%. And so the L allele is making up the difference in those changes in genotype. So the model showed selection for that L allele. So it appeared that the L allele was increasing the number of days to death for the population, which allowed potential growth in the model, at least without hunting. And so what the model suggests is that, if all elk have equal incubation times, as we were showing in the age-specific models, a more negative impact is predicted, similar to what has been published in other models. But if you go back to what I was saying in the beginning of the talk, research from what we've done and in other examples has shown that species have differences in incubation times in relation to their genotype in their response to TSEs in general, which is prion diseases. And again, they are found at lower than expected frequencies in positive samples, indicating that it's likely that these age-specific models aren't probably correct. Just a quick recap for this model portion. In our current hunting scenario, elk were extirpated. In our antlered-only hunting scenario, the population was below objective. In our no hunting scenario, the population became above objective at the end of 100 years. So this suggests that with adaptive management, a level of hunting may exist that is sustainable to a changing population. And the model suggests that elk populations that are exposed to CWD could potentially respond through changes in the frequency of genotypes. And while we know that CWD is negatively impacting wildlife populations, deer and elk. With adaptive management, elk populations may actually be able to adapt to CWD. So how does that relate to the real world? Well, we actually are seeing some selection pressure changing frequencies in certain populations. Robinson et al. Reported an increase in the resistant 9.6 GS genotype for white-tailed deer in Wisconsin. Though that change is pretty small, they do feel that the selective pressure is causing these changes in genotypes. In here, in Wyoming, we are seeing an increase in the F allele in a certain mule deer population, which Dr. DeVivo was going to talk to you a lot about after lunch. However, it seems in other situations in Colorado and elsewhere, they are not reporting these same changes in frequencies and genotypes. And it's unclear why that is, but we know that this disease is chronic in nature, and so it may not be exerting the selective pressure over a short enough time period to see these changes in genotype. So, because it requires a lot of selective pressure and quite a high fitness advantage to see these differences in genotype frequencies. So it may just be that we haven't quite had that pressure to get us where this model is suggesting. So I want to talk to you a little bit about kind of this growing concern about what would happen if we do see this change in genotypes. And the first off is we don't actually know. There's not much data at all out there about how these genotypes might affect the population as a whole. But if CWD is going to exert selective pressure, we would expect to see an increase in the heterozygous genotypes, or those genotypes that offer at least some resistance to CWD. So I wanted to go into what that means in terms of heterozygosity. Well, we know that sexual reproduction, the chief benefit of sexual reproduction is genetic diversification. This gives your offspring the best chance to adapt to a changing environment. This has the potential to mask deleterious genes, and it gives you a wider range of potentially useful genes. And there's a lot of instances where females will actually maximize their heterozygosity through their mate choice. And they do this by selecting males that have greater symmetry, or in the case of birds that have brighter plumage. There are also cases where females select males that have more rare alleles, thereby trying to increase their heterogeneity in their offspring. And then there again are cases where females will mate with multiple mates, again to try to increase that heterozygosity. And this is because, in general, there is no necessarily universal rule, but generally speaking, heterozygosity does confer potential fitness benefits. This is seen through size, health, and symmetry. And there are cases when they've looked at white-tailed deer and correlated increased heterozygosity with fetal growth, greater body size, and antler growth. So again, but what if? I know people still get hung up on the potential fitness disadvantages of this L allele. So if there is a negative downside to the L allele, what we'd expect is in the future is a stability of allele frequencies, with the heterozygote, that MM genotype, being the most common in some unknown makeup of the MM and LL genotypes. And so there's a lot of examples of this in the wild. There's a couple that I just wanted to kind of point at to try to hit this home, I guess. And so one of those is rats and rodenticide. There are populations of rats that have a certain allele that makes them more resistant to rodenticide. And so what you would expect is to see an overabundance of this L allele a higher prevalence of this allele in those rat populations. But instead what you see is a greater number of the heterozygous genotype. And what they believe is that L allele, while beneficial in terms of resistance to rodenticide, it confers a deficiency in vitamin K. And so the rats have kind of, trying to get the benefit of this allele on both sides, they've increased in the heterogeneous genotype. It's a similar scenario if you look at malaria and sickle cell anemia. The allele that is responsible for sickle cell anemia offers resistance or reduced susceptibility to severe malaria. But in these areas where high malaria is, so you see a higher number of heterozygotes, which is kind of a dangerous mix because those that are homozygous for that sickle cell anemia, that tends to be fatal. So there's this balancing act by trying to get the most that you can from these alleles that offer benefits, even if there is a downside to those alleles on another, at least in the homozygous aspect. And so there has also been questions about if we do see these changes in frequencies, won't we be losing diversity? And I guess given the potential selective pressure of CWD, it is likely that we'll see a change in free genotype frequencies, but it is not a heavy enough pressure that the M allele would be lost altogether. And that is all I have for you today. So hopefully we'll get to have lunch earlier, and you can see the teddy bear slide.
What the heck are you doing at Weed and Pest?
Amazing weed and pest work. I work with great people. They pay me well. It's fantastic. Yeah. Yes.
Hi, Amy. In putting your model together, did you consider the Laramie Peak elk herd and the almost 30-year history there, CWD infection for almost 30 years, only 6% infection? The herd has grown fourfold from 2,500 to 10,000, and they winter in very, very large herds. That actual factual on the ground would seem to counteract your computer model of what CWD will do.
Right. So the question, I guess, was saying that so some of the real-world data shows that in areas that have CWD prevalence, this population seems to be doing well, and that is true. So I guess the difference between what we're seeing in some populations currently in comparison to this model is that, again, I wanted to stress that we tried to get a worst-case scenario. So we're giving these animals high levels of prion contamination. Every animal is subjected to this high level of treatment. And this is just an effort to try to get a upper bounds of what CWD infection might look like and how that might impact populations. So it's not necessarily related to what we would actually expect to see. Again, it's hopefully getting at that upper level of exposure. Yes.
Does there seem to be more LL genetic types or MLs or Ms? Which one kind of dominates the pattern?
So the MM is by far the most dominant across any population that's been tested, to my knowledge. Yes. Shane?
Did you run the model at all with some demographic data from, say, Jackson Hole Elk, where they have lower calf survival, increased predation rates?
I did not. There's a couple things I guess I can try to say. So our model had a wide-ranging, calf-to-cow ratio is stochastic, and based on the Pineda elk herd unit, off the top of my head, I think it was roughly 25 to 35 calves per cow, given the time frame that we looked at. And so when we used the sensitivity analysis, that allowed those parameters to go above and below 10%. And so it still didn't have a large impact on the model outcome. With our model assumption, so is that exactly what we'd expect to see in the real world? Not necessarily potentially, but we didn't look at Jack's specific demographics.